This technique allows the combination of the intramuscular injection of the snake venom cardiotoxin with the intramuscular injection of self-delivering siRNA, thereby allowing the analysis of regeneration of skeletal muscle.
The application of self-delivering siRNA is an elegant way to investigate the functional loss of single genes during muscle regeneration, thereby avoiding transgenic animals.
After confirming a lack of response to toe pinch, shave the injection area of the cranial lower leg from the knee to the paw and remove any loose hair. Place the mouse in the supine position on a heating pad covered with a sterile surgical cloth and use 70% ethanol to disinfect the injection area of the cranial lower leg. Next, load an insulin syringe equipped with a 29-gauge needle with 50 microliters of 20 micromolar cardiotoxin and pierce the skin fully into the muscle just distal to the knee. When the needle is in place, inject the cardiotoxin over a 10 to 20 second delivery period along the full length of the muscle while moving the needle back and forth to allow an even distribution of the cardiotoxin, thereby injuring the entire tibialis anterior muscle. Then, return the mouse to its cage on a heating pad with monitoring until full recumbency. 3 days after the surgery, disinfect the injection area as just demonstrated.